Hi everyone, welcome to the Live Longer World podcast. I am your host, Astha Jain. On this podcast, I have conversations with scientists and entrepreneurs that are transforming the field of longevity science. If you wish to stay notified of upcoming podcast releases and sign up for my newsletter, head over to livelongerworld.com. And if you want to receive extensive show notes and transcripts from each episode, you can sign up to be a premium member at livelongerworld.com/premium. My guests today are Dr. Irina and Michael Conboy, who run the Conboy Lab at UC Berkeley. Their research focuses on tissue rejuvenation via plasma dilution and parabiosis. They've pioneered a process called neutral blood exchange, through which they show that they can achieve true rejuvenation in the brain and other tissues of mice to a younger state. This is incredibly exciting, and we dive all into it in today's episode. I hope you enjoy it. Michael and Irina, and welcome to the Live Longer World podcast. It's such a pleasure to have you here today.
Thank you for the invitation.
Yep. Hello. Yeah.
Hello. So you've often spoken of aging as not necessarily something that is an increase in damage with time, but something that our capacity to repair the damage decreases with time. Can you speak about where that concept of aging comes from?
Yeah, sure. Thank you so much for understanding what we usually present or focus on. But I usually give my example, which is comparison between a squirrel and a rat, right? So squirrel and a rat are very similar animals, right? They have similar size, they have similar metabolism, right? A squirrel even has higher metabolism than a rat, and they have similar diet, and they live, of course, in the same entropic environment, which is our planet Earth. But squirrel lives between 20 and 30 years. Some of the squirrels that you guys see and run around are quite old. They might be older than you. Where rat only lives 3 years, only 3 years. So that itself tells us that aging is not simple wear and tear. It is not caused by just tissue damage, but it is a balance between damage and repair. So those of us, and by us, I mean mammals, right, or animals, that can repair our cells and tissue better, live longer.
I see. Okay.
And that is only one example. There are numerous other examples. And many people, some people think that naked mole rat is the extreme example of longevity, but it is not true.
Why is that the case?
Ordinary squirrels that run around most likely outlive naked mole rats. There are numerous other animals that live very long life. I don't know what is the fascination of people with the naked mole rat or how this model became a model, but for looking, at actual scientific results or evidence or experimental hypothesis, naked mole rat really is a colonial kind of exceptional organism, whereas squirrel is kind of like a rat with a bushy tail, but it lives 10 times longer than a rat.
Interesting. I guess you mentioned you don't really know where that comes from then, the model of the naked mole rat. Any conjecture though?
Well, since we're discussing that, there is certain truth about naked mole rat that they long lived, but they are colonial organisms. So not every rat in a colony lives long. It's only, I think, the matriarch lives longer. And there could be numerous reasons why the dominant animal that gets the best of everything would live longer in a colony of animals. In addition, these animals are just so different from many other animals that we know that it would be difficult to say why they live longer. And finally, I don't know if people can actually tag individual naked mole rat animal and prove that that animal actually lived,
Sure
right? I don't know. Yeah. How can you tag them, find them and tag them in the colony? So anyway, this is just, kind of my thoughts about it.
Same way you tag a squirrel.
Same way you tag a squirrel, I guess. Anyways, so so the point of it, not to disprove of my introduction, is not to disprove at all that naked mole rat is good model, it might be excellent model, but just to say that it is not an exceptional model. There are other animals that we know much better that live very long. And then there are other animals that we also know very well, like rats, and they live only 3 years. So it cannot happen simply from tissue damage.
Okay. And then you think it's basically a capacity to repair the damage that is possibly causing aging at different rates.
That's what we think in our lab.
Okay. So I want to jump into some of your work around neutral blood exchange. But before we get to that, I think there were some experiments that you did around heterochronic parabiosis that led to your work on neutral blood exchange. So perhaps can you explain what heterochronic parabiosis is and what were the studies around that?
25 years ago, we had discussions about aging and why we age, and rejuvenation and so on and so forth. And one of the things that we discussed, and we're probably not the only one to discuss this, is why is it that when you age, all the tissues and organs tend to age together, right? It's almost an exception when you see someone who's older that has particularly young looking hair or skin or eyes, or maybe they're still very fit or something like that. Usually everything's kind of everything's declining together, right? So we had this hypothesis that maybe there's something that connects all the tissues and coordinates, whether it's a tissue that's on the outside, or in the lung epithelium, like epithelium that gets a lot of oxygen, or if it's on the inside, if it's high metabolic or low metabolic, if it's derived from different embryonic development layers, right? The inside, the outside, and the middle. That all these tissues, there's something that coordinates it. So we said, okay, what could coordinate? The nervous system innervates everywhere. The blood flows pretty much everywhere, and then the blood vasculature carries the blood everywhere. So our thought was, well, this would be interesting, and if we could just do an experiment to test it, maybe we could transplant the nervous system from a young animal to the old animal. But we didn't know anyone who could do that, right? Or we'll transplant the vasculature system from a young animal to an old animal. No, we don't know how to do that. But blood was something that we thought we could do. So it was sort of on a thought to do someday is, well, if we were working in a lab with mice at the time. If we get to work with something a little bigger that has bigger veins, maybe we could do a transfusion between a young, say young rat and an old rat. And that kind of, got forgotten or put on the back burner. And then in 2003 or so, 2002, 2003, Irina presented a paper in Journal Club, and we were postdocs together in Tom Rando's lab at Stanford. And she described an experiment that was done by another lab at Stanford, in Irv Weissman's lab, where they were looking at can stem cells from one tissue type, in this case it was the blood, can they contribute to other tissues like nervous system or muscle or anything like that? And at the time, that was a question. And to answer that question, what they used was this technique called parabiosis. They took one mouse that was transgenic for green fluorescence, so all of its cells are green, and stitched it to another animal that didn't have any green cells. It was wild type. And what happens is you cut down one side of one animal and the mirror image down the other side of the other animal. And instead of closing the skin on the same animal, you stitch it across the two animals. And when the skin grows together, the blood vessels in the skin also grow together and merge. And a few weeks after stitching them together, you've created sort of like a Siamese twin between the two animals, and blood will flow from one animal to the other. And the cells in the blood will go from one animal to the other. And they use that to determine that the blood cells did not contribute to anything else besides blood.
Yeah.
Right. And so as she's giving this talk, I'm sitting in the back of the conference room. I had probably had my cookies and cookies and tea, and I'm like sort of dozing off. And all of a sudden I woke up and realized that, wait a minute, this technique that they used, we could answer the question about the blood with, because you don't have to do any vascular surgery, right? And mice have really small blood vessels. I mean, there's just technically, it's very difficult to get a tube in there and then to pump from one animal to the other blood, right? So, but this technique it's gross, but it's sort of simple, right? It's almost, you know, Frankensteinian in its right? So we,
and by that time we already did experiments with cells and cultures. So we would take young stem cells and put them in old blood serum, and they would immediately age. They behave like old stem cells in a matter of hours. So that was curious. And then if you put old stem cells in a young blood serum, they would have rejuvenative responses. They would divide better and would differentiate into muscle better. So we had already this idea that aging is systemic and rejuvenation is systemic, but we didn't have any way to prove it in live animals until Mike had this breakthrough in the Journal Club presentation. And then Irv Weissman, whose laboratory published those seminal cell papers on parabiosis between two young mice. They connected one young mouse with another young mouse. He was my thesis committee member. And so I knew him. He's super famous, really remarkable scientist. And I knew him for all of the 5 years of my PhD, where we had to meet with my thesis committee like regularly, every couple of times a semester. And Irv was always fly fishing in Patagonia or doing other things that was difficult to pinpoint him, but eventually he would show up at the meeting and give me the most brilliant advice. So then I basically set up a short meeting with Irv. I came to his office and I said, hey, we discussed this Journal Club, your paper, and Mike, he knew Mike as well, has this idea, what if we connect young mouse with an old mouse? And even before I really finished the sentence, he said, this is great. Here is the person in my lab, postdoc Amy Wagers, and she will teach you how to connect mice together. And then Amy basically taught us how to do it. And then that's how she became interested in aging and rejuvenation. And she learned about muscle approach and stem cells and muscles from Tom Rando and from us. And that's spearheaded her career in that direction.
That's fantastic. So what were the results of the study then?
So basically we looked at 3 different tissues. Looked at brain, liver, and muscle. And in the old animal, all 3 tissues seemed to be rejuvenated. They had improved parameters. Whatever we looked at was better. And the young animals showed some decrease in performance of those 3 tissues, right? We didn't see as many new neurons being born in the brain. We didn't see such good muscle regeneration. And the livers started to, saw a decrease in
regeneration.
Yeah, well, the cells that are sort of maintaining the health of the liver. And we've seen that sort of again and again and again as we looked from parabiosis to blood exchange, and then to the sort of latest tack we're on, which is the neutral blood. And at the time when we published that, we didn't know whether it was good stuff in the young blood or bad stuff in the old blood. And I remember having discussions with Tom Rando about this when we were writing the paper, right? And I think Irina might have been, there's bad stuff, thinking that there's bad stuff in old blood, and I was thinking, well, there's probably good stuff in young blood. And Tom was agnostic. He was like, well, we don't know, right? So so when we were writing the paper, we were careful about that, right? So
but it didn't help because everybody just decided that there is good stuff in the young blood, and young blood could be medicine.
Not everybody, we didn't, right? Yeah.
Most people, most subsequent studies were focused on that, unfortunately, because not only that is not true, but it is just overly simplistic. And intuitive human thinking very often is completely different from how biology works.
Well, I'm gonna qualify that, say that there is good stuff in young blood. And as we others have found, there's definitely bad stuff in old blood. The question is, if you start thinking now about going from experiments in the lab to something you want to maybe try on people, what's the best approach? So
yeah, and so also from my perspective, good stuff in young blood is not going to work in the presence of bad stuff in the old blood. And we actually have proven it several times. So for example, if you have a young animal and you exchange it with 50% of old blood, right? And we can do these experiments in mice only because each mouse is like a identical twin of another mouse. So they all have exactly the same genetic background. So we can exchange as much blood as we want without any problem with immune rejection or immune response, right? So then if you think about it, you have a young mouse, right? And that young mouse has all of the young cells and tissues. So long telomeres, no reactive oxygen species damage, no DNA damage. Everything is young. And it has 50% of its young blood, 50%. Mm-hmm. But exchanging it once with 50% of old blood instantaneously ages it. And then provides this huge question mark. What will happen if you have an old person with everything old and 100% of old blood and you just inject a little bit of the young stuff?
The popular press basically picked up on that. It's just simple blood transfusion. And it sounds like, yes, there might be good stuff in young blood, but simple blood transfusion from young to old doesn't have all the benefits without suturing. Is that correct?
Okay, so the suturing, right? So the first test we did was the suturing because that's what we figured out what we could do. It turns out that you actually can do vascular surgery on mice. We just didn't know how to do it. And after we published the parabiosis work, then Irina got her position up at UC Berkeley, Cal Berkeley, and we were doing experiments there. And one of the kind of the Illuminati in the aging field, Aubrey de Grey used to have discussions with us about aging and experiments they were doing, and he wasn't the only one who was asking this question, but he kept pushing it. How do we know that it's the blood that had the rejuvenating factors as opposed to the young animal system that this old animal was using, basically, right? So like the insulin, glucose regulation system, the digestive system of the young animal, the kidneys, the liver, the filtration system, just being physically moved, the heart physically moved around the cage more because the young animal is more active. How do we know that wasn't what was rejuvenating the old animal and not the blood? And we knew this, and we had talked about this, that it was, the test would be that no, we have to just limit it to just blood, just do a transfusion. So Aubrey found he knew someone who had the skills to do vascular surgery on mice, and he also had a bit of money to give the lab to do that work. So he introduced us to Dr. Justin Rebo, who came on board and did that set of experiments with an undergrad in the lab, Milad Medapur. And the two of those published, there's a few other people in that paper too, but they did most of the work publishing that paper on blood exchange. And that was surprising, that one simple exchange back and forth between a young animal and old animal. So, it takes maybe half an hour to do the exchange. Then you unplug them, they go their separate ways. Look a few days later at the old animal, and again, the brain is rejuvenated, the liver is rejuvenated.
The brain is not rejuvenated
a little bit, not right? At all. There was a trend, right?
The brain is not rejuvenated at all. The liver and muscle are rejuvenated.
Okay.
We argue about this a lot. Let the audience go look at the paper, and there's a trend there.
Very good.
All right. So you can argue maybe it's not as dramatic as with the parabiosis, or later on we did with the neutral blood exchange, right? But in general, it was rejuvenative to the old animal, and like we saw with the parabiosis, it was detrimental to the young animal.
Going into neutral blood exchange, then you mentioned, you had this question: was it the good factors in the young blood, or was it the bad factors in the old blood? And I think that's what led you all to do the following experiments, which you all published in your 2020 paper and introduced the idea of neutral blood exchange. So I have a couple of questions there, but maybe you can start with what the study was, what you were controlling for, and what is even neutral blood exchange.
Right. So the question there was we would argue that whether there's, good stuff in the young blood or bad stuff in the old blood. And we said, well, what if we could exchange with some blood that didn't have any factors, that was neither young nor old, right? Then would the old animal, from losing its old factors in this situation, would it be rejuvenated, or would there be no change? Meaning that it's not the dilution of old factors that improves. We argued around the lab meeting and what would be sort of a neutral age blood. And we were also talking at the time as well, what would be the component of the blood that has the rejuvenating activity? Is it the cells, is the red blood cells, the white blood cells, is it the plasma, is it something in the plasma, right? So we just had sort of technically sort of a simple experiment and said, well, we'll spin out the cells, the red blood cells and the white blood cells, and throw away the plasma, and then resuspend those cells in saline, purified saline with purified albumin protein. So it really doesn't have, the plasma part then is replaced by something that really doesn't have any age. It's just purified, salts and water and protein. And so we did that, and that probably has given some of the more surprising results so far, is that was dramatically rejuvenative. And there's obviously there's no young factors in there at all. It's just it's getting rid of old factors, and not even surprisingly, it's not like we're getting rid of 99% of the old factors. It's just decreasing them, all of them across the board, by a significant amount, like half. So we're still trying to figure out what the factors are, combinations and how they affect the cells in the tissues that respond to that. But it definitely seems to definitely shows that there's something in the old blood that if you could just kind of clean it up and do a plasma change, like an oil change for your car, that things run a lot better after that, right? Things are improved in the old animal, and quite possibly old people too, because this is a technique that actually exists for people, people that have they're experiencing organ rejection, or they have like liver failure or something like that. You can do this sort of blood purification. You throw away the contaminants in the floating around in the blood and replace it by purified saline and albumin. It for those people, it saves their life. If you're at age-related diseases, maybe this will be something that can kind of help restore the system.
I would like to clarify that in the heterochronic blood exchange, where in which young mice are exchanged with old blood, old mice are exchanged with young blood, or we have controls, right? Young mice are exchanged with young blood and old mice are exchanged with old blood. So those are controls for the process. The most profound effect that is right in your face is that young mice become much worse in their brain health and regeneration. So basically, a single dose of old blood obliterates formation of new neurons in their brain, and they become less cognitively capable. They do not perform well in tests that measure short-term memory. Old mice, based on our data, do not become statistically better from young blood. Mike mentioned that there might be a trend, which is possible, but there is no statistical significance in their improvements.
I see. Okay. But there was some improvement in liver and muscle.
And definitely there was a statistically significant improvement in their liver and muscle. So there is something about the brain that is really progerotic, and it's easy to age it by old factors, even if it is healthy, brand new brain, and you still have 50% of young blood still that it ages. So that's quite interesting, right? Because there are several companies and trials which try to inject people who have Alzheimer's disease and are old people with young plasma. And as far as I understand, the trial was not really focused on determining efficacy, just determining safety, but they did not observe any improvements. So they did not observe any improvements, and that observation is consistent with our research.
Thanks for clarifying that. That's super interesting.
I always give this, metaphor that I heard it someplace, I don't know where, that if you have a bunch of rotten bananas and you throw one good banana in that bunch, what do you think will happen?
Oh, yeah. All the good
The
bananas will
rotten
be
banana
rotten.
turn good, right? No. Will
Mhm.
that one good banana become rotten? Most likely. So that's my metaphor of that whole phenomenon.
Yeah, that's a great one, actually. So on the neutral blood exchange part, Mike, you mentioned that it was dramatically rejuvenative. And I think y'all have noted positive effects in liver, muscle, cell division, and neurogenesis, neuroinflammation as well. Can you speak to some of these positive effects that you saw with neutral blood exchange?
So what we observed was, really short of remarkable, is that hippocampal neurogenesis became not just better, but as good as in the young animals. So there was no statistically significant difference now between old mouse that was exchanged with neutral blood exchange and the young control animal. And the same was true for the muscle regeneration after injury, reduction of muscle fibrosis, and also kind of a health of newly regenerated muscle fibers. The same was also true for reduction of liver adiposity and fibrosis. And then in subsequent studies, we determined that neuroinflammation declined and senescence of brain also declined. And what is interesting is that that was true rejuvenation, right? It is not something that was just slightly better. In many studies, people measure, for example, life extension of animals, and they measure other parameters where they say, oh, now there is 10% improvement or 20% improvement. And that is already significant or important. In our situation, it was true rejuvenation. In everything that we studied, there was no difference, no longer a difference between old tissue that was rejuvenated and the control young tissue.
How long do you think these effects last?
We don't know. So that needs to be studied, and it could be studied in mice, right? But ideally we would study it in people. So we are conducting clinical studies with Dr. Dobry Kiprov, because as Mike mentioned, this procedure is FDA approved, and Dr. Kiprov can prescribe it to different patients who do not have any serious illness. So they do not have, terminal Alzheimer's or cancer. But as many people, when they get older, they just have a little ailments, things are not perfect anymore. So based on his, clinical discretion, he prescribes those, and then we get blood samples. And so from blood samples, by analyzing various other things, hundreds of proteins and gene expression and so forth, we can then understand how long the effects last. So that study is just ongoing right now.
Working on pilot trial, right? So it'd be cheaper, and then you can, say, okay, give a number of patients, each of them had their individual ailments. What happens to the patients after a round of this, a couple rounds, 6 months later, a year later, how are their ailments doing?
Follow-up visits. Stuff like
if I had to guess, I would say that this kind of thing should last a month at least. I'd be surprised if it lasts longer than a year. But if you had a few rounds of this and got perked up the way the mice get perked up, whereas maybe preliminary reports from people that seem to be doing better, that might be enough to allow a patient to change the rest of their lifestyle, to now start being more active and improving their diet and whatever other things that generally tend to help longevity and health span and things like that. But that can be difficult when there's a particular ailment or two that are just keeping you from moving or eating well, or whatever.
Yeah, and I has a good term for that. It's called comorbidities of aging, right? Comorbidities. That each one thing is perhaps manageable, but when you have several of them, that cause profound health decline. So if therapeutic plasmapheresis reduces comorbidities of aging, then other therapies or even lifestyle changes are expected to have a bigger positive impact.
Absolutely. Yeah. So this pilot project, is it ongoing right now?
Yes.
Yeah, Kiprov is performing these procedures.
Fantastic. How do you think neutral blood exchange is working mechanistically? I guess it's diluting some of the age-elevated proteins. I've heard previously you've mentioned it's probably working in 2 waves of rejuvenation. Maybe if you can talk about the 2 waves, or if you have further information on that, that would be helpful to know as well.
It could be my turn, but I don't know what the 2 waves are. I don't remember talking about waves.
Yeah, so no, I will start with 2 waves. So and then we'll go turn.
Okay.
So basically, the way I describe it is that the first wave simply comes from the diminished inflammatory proteins that are present in our blood. So those proteins by themselves really are vital for our health at the young healthy levels. We cannot really throw them out. But when we become older, the levels become elevated by 3, 5-fold, and then they start working counterproductively, inhibiting tissue repair instead of activating it. So that is the first wave. And then once those are removed, the second wave comes from changes in gene expression of so-called young proteins, which are diminished in our blood. And they are diminished because they were actually suppressed at the level of regulation of gene expression by those age-elevated proteins. So then sometime after the procedure, then all of the young proteins are elevated. You don't need to add them back ectopically. You don't need young blood anymore.
And whether that means youth, as in, like it was when you're younger, or whether that means healthy aging, meaning that you're still old, but everything's working better now. We don't know that yet. So we're still studying that. And we don't know how this is going to play out as far as, health span and lifespan. We would study mice for that until some clinical trial result comes in. But until, in the absence of that, our lab is a experimental lab. So we study what goes on in the mice. So we'll see what happens with that. But that, again, the results of that in either case would be several years out.
Yeah, we study what's going on in mice and what's going on with human cells. That's what we study. And then we are expanding actually our network of collaborations with clinicians. So we collaborate with Dobry, who is our most favorite collaborator clinician. Then other people start to approach us, and they are located in various parts of US, and they also are physicians, and they ask if we would like to expand collaborations with them. So the hope is that by doing the studies and also analyzing the big data that is produced in the studies, we would be able to identify which are specific proteins that we diminish and which are specific proteins that become elevated, positively elevated. So then we can make therapeutic plasmapheresis a better procedure, so the effects will last longer. And then eventually phase it out. So people do not even need to have blood exchange with saline albumin. They could simply have a cocktail of designer pharmaceuticals that would do the same.
Interesting. The point on specific proteins makes me think that I believe you mentioned that there are probably small number of critical factors in the blood that are more negative than the others, or contributing to, call it, aging. Have you been able to identify what these small number of critical factors are that are more negative?
Yeah. So first of all, yes, it's a very good understanding, right? People always think that there are hundreds or thousands of changes and we need to account for all of them. But in reality, you're completely correct. There are few critical proteins. And if you normalize their levels, everything else falls into place, right? I give this another metaphor by saying that we have an army, but the army is commanded by generals. And if you simply then convince the general what to do, the soldiers will follow. So it is very, very similar situation. We have hierarchy of proteins and they all interact with each other, but in a predictable way, such as only a few of them need to be changed or normalized. With respect to specific candidates, we identified one of the most important and the critical, or one of the most important and critical, which is called TGF-beta, transforming growth factor beta. And we published that simply attenuating TGF-beta alone, or in combination with increasing the useful protein that declines, peptide in fact, called oxytocin, will recapitulate many of the positive effects of neutral blood exchange or parabiosis. But this combination, while promising, is not as strong as the diluting of plasma through plasmapheresis. So it works, right? But it doesn't make all tissue just identical to young, which suggests that 2 of them are not enough, but we need to identify more of them. So perhaps 3, 4, 5, then we will be on the same page as, as blood dilution.
As blood dilution. Interesting. How do you inhibit TGF-beta 1?
So we inhibit TGF-beta signaling pathway, TGF-beta receptor, by a small molecule. We actually published on that, right? So we published on that in several papers, I think 3 papers in the past, maybe 5 years. And there is a small molecule which binds to the receptor of TGF-beta. And it's not just TGF-beta 1, it's also myostatin and GDF and other proteins that bind to this receptor. And then it inhibits the kinase activity of the receptor. So even though the ligand is present, the signaling is absent. I don't know if it is over the top for the people that listen to podcast, but again, to give another metaphor, if you go outside and it's very, very sunny, you cannot prevent sun from shining, right? But you can put on sunglasses. And then, even if there's a lot of UV, they do not reach your, your eyes. So, so the approach is, metaphorically similar. We do not really prevent the levels of TGF-beta, but we prevent TGF-beta from signaling to cells.
Interesting. I see. Is it, I guess, is this something that gets elevated more with age? Is it useful when we're younger? What is the function that it serves?
It's definitely elevated with age. So as all of there is no protein or cell or gene that evolved to make us old or sick, right? Those do not exist. So when people tell about senescent cells or P16 or anything, it all means something only in relative terms. So TGF-beta at young levels is indispensable for us to be alive. It controls normal cell responses. It also regulates immune responses. It functions everywhere. But
embryogenesis,
even for us to develop and be born, right? Exactly. But then when we grow old, the levels go up in blood circulation and in tissues by several fold, by like 3 to 5 fold. And at those levels, TGF-beta signaling becomes counterproductive. It kind of hijacks other cell networks and signaling just to make more TGF-beta, and it does horrible things like promoting fibrosis and inflammation.
Interesting. Yeah. So I have a few other questions on neutral blood exchange. One was that you compared it to Senolytics as well. What were some of those findings there? And also as a follow-up, did you compare to any other interventions as well?
Yeah, so the work on comparison between neutral blood exchange and Senolytics is actually ongoing. It's a collaborative work with Professor Judy Campisi, who is the guru. She uncovered really the presence of senescent cells and how to measure them. She is my scientific idol, pretty much. So we collaborate with her on that. And with her former postdoc, a brilliant young woman scientist, Oki Jeon, who just started her laboratory in Korea. So we are exploring what are the similarities and differences between the effects of plasma dilution or blood exchange and Senolytics. That is very, very interesting, and everybody is asking us what is going to happen. And so the manuscript is under review, and hopefully soon it will be published, and you guys will know what happens there. We don't know. With respect to some other approaches, people use metabolic inhibitors, for example, metformin. They use growth hormone in various combinations with various things, such as metformin and other things. They also use NAD, which changes metabolism. They also try to use young plasma factors and young plasma from young blood. But what I wanted to mention is that the rejuvenation by dilution is, as far as I know, the only approach that is true rejuvenation, where all tissue becomes young. In all other approaches, there are some improvements, and various laboratories report different successes. And that is all great because, as I mentioned, aging is multifactorial, complex phenomena, so we all need to work together to understand it completely. But we think that we really are tapping on the door of the actual cause of aging, because old tissue very quickly after one half-an-hour procedure becomes indistinguishable from young tissue. So therefore, looking at it as a process, yes, we found the key to the process. Now we still do not understand all the mechanisms, and that is the next step. How does such robust rapid rejuvenation take place? It is very surprising. So now we are looking into that.
Yeah, which is incredible, and makes me ask you, what are some other questions that are unanswered or you're looking into on neutral blood exchange?
Yeah, I don't know how, this is a very big question, right? What are the small that are unanswered? I think that whenever we make a step towards understanding something, we realize that horizon becomes bigger, right? And there are many more that we do not understand. So those are all of the questions here. Is that, namely, is that first of all, how did such a process evolve? Why would you have accumulation of the negative factors in blood, in your circulation, that would broadly make all of your tissues progesteronic? Mm-hmm. What is the evolutionary significance? What is the conservation between mice and people? And then once we understand that, what is the fundamental paradigm of that, then the question is translational. What are the key steps, and can we then reverse engineer them to our advantage so we actually develop meaningful anti-aging pharmacological approach? And even though it seems maybe far-fetched, but I would like to mention that just less than 200 years ago, when there was no antibiotics, many diseases were not only incurable but impossible to imagine that we will cure them. Mm-hmm. And then pretty much just with one class of drugs, is just antibiotics, something that distinguishes bacteria from our eukaryotic cells. Then all of those diseases became curable as a class, right, as bacterial disease, right? So I always think about aging in the same way, is that we need antibiotic against aging. We need something that will not be just curing, Alzheimer's disease separately from Parkinson's disease and separately from osteoporosis, but will cure disease of aging as a class. So I guess to answer your question in another very broad answer, this is what we are looking for, an antibiotic against aging.
No, that's fantastic. I'm totally on board, and I really hope we solve some of these questions, answer some of these questions quite soon. You already mentioned oxytocin and inhibiting TGF-beta 1, but I wanted to go back and ask you, why do you think that's working on the oxytocin side, where I guess increasing oxytocin levels can rejuvenate muscle function? Why do you think, what's happening there mechanistically?
So what happens, we actually know what happens. And we started this discovery back in 2003 when Mike and I were in Tom Randall laboratory, or 2002 even, when we understood that Notch signaling is the key to activation of muscle stem cells. And with age, Notch signaling declines because Notch ligand delta fails to be expressed on the surface of the muscle fibers.
What is Notch signaling?
Right. So Notch is an evolutionary conserved signal transduction pathway by which cell communicate with other cell, which exists in fruit flies and in worms, and also in mice and in people. So without that pathway, we would not be born. Any disturbance, significant disturbance in this pathway, and there is no embryonic development. And then without this pathway, we would not be able to repair our organs and tissues, because what was tested when we were developing is then utilized again when we regenerate. So, you find something good and then you use it again when you need to repair the tissue, right? So you build the tissue using Notch pathway and then you repair it using Notch pathway. Mm-hmm. So Notch pathway is activated by its ligand on the surface of adjacent cell. And so Notch itself, which is a receptor protein on the cell surface, is present on the muscle stem cell. Muscle stem cell sits right on top of the muscle fiber. And when muscle fiber is injured, it upregulates Notch ligand delta, therefore activating stem cells in the vicinity of the injury. So it's almost like a positional, not almost like, it is a positional cue. I am injured here, so you need to make more muscle right here where there is injury. So it's a very, very beautiful, elegant system.
They have these descriptive names because these genes were just discovered and first characterized in some
fruit fly.
Yeah, some simple experimental organism like fruit flies, and that reflects a phenotype, the shape of the wing or whatever. That
the wings had notched.
Right.
Right. Yeah, exactly. That's all the most of the names of the genes come from.
And we're talking about the hundreds of proteins that are in blood, but that there's only maybe a handful of signaling pathways that the cells respond to, and the Notch pathway is one of those, right?
Yes.
These growth factor pathways would be another one. So you have more growth factors that signal cells to produce more of the ligand for Notch, the key for the Notch lock, which would be the delta signal,
delta,
right? So the pathways that kind of intertwine together, and the stem cells need to get these signals in order to wake up and start dividing, to make more progenitors, to make more differentiated cells, to make more tissue.
Okay. So then what happens with aging? So we published a developmental cell paper with Tom Rando in 2002, describing Notch as the key regulator of adult muscle repair. And then in 2003, we published science paper also in Tom Rando lab, saying that with age, Notch ligand delta fails to be expressed on the injured fiber. And so old muscle stem cells in the old muscle just fails to wake up and make the new muscle fiber. So then later on, again, based on the evolutionary conservation and the papers that were published by the field in fruit flies and worms, delta is controlled by MAP kinase signaling, which is the growth factor pathway. And oxytocin is one of the factors that is present in blood and activates MAP kinase signaling. So if you are now an old animal or person, and now with age, oxytocin declines, so there is no MAP kinase, there is no delta, Notch is not active, and stem cells remain sleeping, and muscle remains unrepaired. You can trigger this whole system by adding oxytocin, which will then induce more delta, Notch will be activated, and then muscle is in a better shape.
Oh, that's very interesting. Thanks for the explanation. That made it easy to understand.
And this is one kind of logical process or network out of several canonical ones that we need to restore to maintain our health and youth.
Yes, as you've said, aging is complicated. Lots going on. So if we can now fix a lot of these systems, I'm sure. A lighter question to end with. I believe y'all have chickens or used to have chickens, and and also ravens to babysit them. Is that true?
What was it? So the ravens to babysit them? Well, ravens are not going to do anything, right? So, but there are some ravens that seem to like to hang out on the hill, maybe nest in the hill behind our house here. So we have tried to encourage them to visit the yard, because the more they like to visit the yard, the more they like to defend their territory against chicken hawks.
Is there anything else about your research or neutral blood exchange or anything else on aging that I may have missed and you wanted to cover?
I don't think so. I just would like to thank you again for allowing us this opportunity to speak and for very, very thorough podcast.
Hi again, everyone. I hope you enjoyed the episode and learned from it. If so, please show some support by subscribing to my YouTube channel, Live Longer World, liking the video, sharing the podcast, and leaving a review on Apple Podcasts. If you want to dive into these topics further and receive extensive show notes and transcripts from each episode, you can sign up to be a premium subscriber at livelongerworld.com/premium. To be notified of upcoming podcast releases, Sign up on my website at livelongerworld.com. Follow me on Twitter at livelongerworld and Instagram at longevityfuture. Thanks for listening, stay in good health, and I'll catch you soon.